Hey, Dr. Jared Pelo. I'm really excited to introduce my guest today, Matt Yousefzadeh. He's an amazing guy. He's really fun to talk to, all about aging. He's a great scientist. He does really good work, specializing around senolytics and biomarkers of aging and all sorts of aging related knowledge. You can follow him on X, formerly known as Twitter, where he's quite active. And I hope you enjoy our discussion. All right, Matt, thanks for making time. Why don't you just start out, give a quick introduction of yourself?
Yeah, thanks, Jared. Thanks for having me today. So I'm Matt Yousefzadeh. I'm a faculty member at Columbia University Medical Center in the Center for Translational Immunology, as well as the Center for Healthy Longevity. And my lab really works on the intersection of aging and immunity, as well as looking at biomarkers of aging.
Awesome. I think to get started, I'd love just to hear your story. Like, how did you end up deciding you wanted to study aging?
Yeah, so I actually came before I got into aging. I was really working in cancer and especially looking at DNA damage and DNA repair and lack of DNA repair, how that can drive cancer. But as I got into my postdoctoral fellowship, I transitioned over to aging, and we really looked at how DNA damage or the lack of efficient repair of DNA damage can also drive aging as well. So there's a lot of intersections between aging and cancer.
Did you always know you wanted to be a basic researcher? Did you grow up like playing with chemistry labs at home and like knowing you wanted to go into science?
No, I wanted to be a professional baseball player, but neither I could throw a curveball or hit a curveball, so that kind of went away. And I figured I need to do something with my life. And in college, I thought I wanted to study business and transitioned over to biology because I had a grandmother when I was in college who got sick and died of cancer. And when I was a graduate student, I went over to aging because I watched my mom, who was really sort of this healthy, vibrant person. When I started graduate school, she passed away when I was in graduate school, but I noticed that she started to accumulate a number of these sort of age-related deficits. And so, yeah, I would come home at Christmas, and the next time I'd come home, she'd look even older. And she passed away at 53, and so that really kind of got me interested in aging.
Yeah, it's fascinating. I feel like everybody I meet that works in this field, like we all have some sort of story about, like we were affected by aging, by disease, by all of these different things. And it sounds like you had both, cancer and then aging. So yeah, I'm excited to dig in with you. So, you mentioned one of your focuses is biomarkers. And biomarkers are super interesting to me because I'm trying to know, like, if I prescribe something, is it doing any good? If I have somebody change a habit, are we making any progress? And so I'd love to hear your thoughts on kind of how you think about biomarkers around age-related disease or aging and kind of go from there.
Yeah, I think a lot of people look at biomarkers either it's sort of a predictive pattern where, what's your chance of morbidity or mortality? Other people, like I am, we're looking at interventions, and are these interventions responsive? And we're trying to use biomarkers to study that, right? And so I think that sort of fits with a lot of these wellness clinics of, are these things that you're doing, are they making meaningful impacts? And really, kind of biomarkers fall into a couple of different buckets. It could be everything from sort of the odometer of how many miles you have on your body, the speedometer, right? How fast or how slow are you aging? Or, is it also the doomsday clock of how much time do you have left?
Yeah, I mean, it's fascinating to me. So every day in my kind of like my Google News feed, like I just see so much news around like these, new study shows, X supplement does Y thing, or, we know from multiple kind of predictive models about cardiovascular risk, and that's mostly all driven from a biomarker. It's either LDL or
Mhm.
ApoB. So I'm curious, when you think about kind of biomarkers around aging, like how do you think about that? Do you bucket those in any specific way? Which ones are interesting to you?
Yeah, I think broadly, biomarkers for me fall into sort of three different categories. You have a molecular, like you say, it could be your LDL, HDL. For me, it could be some specific molecule or analyte that we're looking for. Another one is physiological. So that could be something from a metabolic panel. It could be also uh, your VO2 max, your resting metabolic rate, something that's muscle mass. And then there's also the realm of digital, right? Things that you get from wearables that people wear, smart watches, or they're getting functional MRIs or different body imaging modalities done.
Yeah.
And so there's really sort of broadly, I think, those three different categories. But for us, we're really interested in these things called the hallmarks of aging, where we know that aging is really the root cause of many different diseases. Like say cardiovascular disease, sort of hypertension, bad diet, everything. Those are really strong risk factors. But if you were to sort of look at them in comparison to aging, aging trumps it all of those factors. And so what's been found is there's a number of these different molecular drivers of aging that we sort of termed either the pillars or hallmarks of aging. And so I think from the biomarker perspective, it's really interesting to sort of investigate those in sort of health and disease, as well as in response to these intervention studies, and either sort of a pre or post measure for the intervention of, did your health get better, but did these biomarkers also get better? And that will allow us to determine, what's the validity of these biomarkers? Are they behaving like they should as well?
Right. Now, I know there's, 9 to 12 hallmarks of aging. Is like, I think we just updated it to 12 this year. Um, so as you think about kind of in your lab, are there any of those hallmarks or pillars of aging that like you're specifically kind of looking at?
So my lab is really interested in cellular senescence, and it's a state where cells can become stressed out. It could be through damage or some other endogenous or exogenous source of stress. And what they do is they no longer proliferate. It's a really an evolved anti-cancer response, but these cells sort of stick around throughout age. They're really resistant to cell death, and they're metabolically active. So they produce a lot of inflammatory material that contributes to this state of inflammatory aging that's known as inflammaging. And so what we like to do is survey for those cells or the inflammatory factors that they're producing. Another thing, we like to look at DNA damage. We know that tracks with aging. People that have DNA repair deficiencies, they do have typically a predisposition to cancer, but they also show a myriad of age-related effects.
Mhm.
And from there, we like to sort of compare it to some of the functional metrics they have, either physical function, which we've done before with a group at the Mayo Clinic, or in this case, we're looking at liver transplant patients and looking at frailty.
Mm. You said a couple things that I think are worth going a little bit deeper on. You kind of explained that senescent cells, like there's a reason the body allows
Mhm.
senescent cells to happen. And would you kind of explain that a little bit more? Because I think sometimes people are just
Sure.
like, you get old, your cells turn
Mhm.
into these zombie cells, but like there's a reason that that happens. So, or at least we believe we kind of understand a reason that our body allows it to happen. So I'd love for you to explain that a little more.
Yeah, so basically, our cells accumulate damage with time. There's all these theories of aging, but one thing they share in common is the accumulation of molecular damage. And so that could be damaged proteins, damaged lipids, or nucleic acids. And eventually, over time, when a cell accumulates enough damage, it can either transform into a cancer cell, it can undergo programmed cell death in the form of apoptosis or another programmed cell death pathway, or it could possibly go into a state of cellular senescence where the cell, it no longer proliferates. It's typically stably arrested, and it doesn't matter how much good stuff you give it to try to make it proliferate, it won't do it.
Hmm.
And what we knew for the longest time, we could see these things, they would occur, but we just thought that these were these little funny cells that sort of accumulated with aging. We thought they were just sort of byproducts. But starting about a little over a decade ago, there were some really seminal studies that showed they were actually causal for aging, that they themselves could be pathogenic drivers of aging.
Mm.
And so there's really a strong role now for senescent cells and the accumulation of them in a number of different chronic age-related conditions, as well as just general aging.
Yeah, that's interesting. So these senescent cells are somewhat protective of like moving on to cancer. So
Hmm.
they keep the cell from moving on to cancer. But then the accumulation of these senescent cells increase the inflammation in your body. We know that they kind of take over where you wouldn't have as many healthy cells. Like we see this in the immune system, right?
Yep.
We'll see this senescent load. And how can we think about how does the body normally clear out senescent cells?
That's a really good point. So, going back to them really quickly, senescent cells are, we use the analogy that the bad apple can spoil the bushel. And that's what a senescent cell can do with sort of the surrounding tissues by producing localized inflammation that either make the cells around it senescent, or it damages them and reduces the ability of your body to regenerate tissues, or just disrupts normal function. So the immune system is sort of tasked with clearing out these senescent cells. So not only as you get older, are you making more of these senescent cells, but the immune system can also accumulate senescent cells as well too, and no longer do their job as effectively clearing it. So not only are you making more trash, but you're losing the ability to take the trash out.
Right. And what's always whenever I talk to anybody about aging, what's remarkable to me is so when you start digging into senescent cells, like we can simplify it as much as we want, but it's actually pretty complex. And when you start digging into it, you realize that's just one of the hallmarks, right? You've
Yes.
got 11 more that are just as complex. And, like I'll hear people talk about, the different theories, the information theory of aging
Mhm.
or whatever it is, which it's really nice to try to simplify it and say, well, it's like a CD getting scratched and you want to buff
Mhm.
out the scratches, but it's really complex. Um, so yeah, I'm just curious, like, how do you think about it? Do you like to simplify it, or do you like to more lean into like this is complex? We're gonna have to solve this like a little bit at a time.
I think I think you have to approach it from both ways. You have to recognize the complexity and that these hallmarks are all interrelated, sort of poking one pokes the other,
Hmm.
or it pokes all of them, but just at different stages and how much. But at some point, to really start to approach it, you kind of do have to take a reductionist view, and you have to start somewhere, right? Even if it's a flawed attempt. And so what you're trying to do is just build a model of what's going on, and just through sort of iterative approaches, you're just refining your model, figuring out, okay, this holds up, this doesn't hold up. What's the effect of senescence on autophagy, sort of the recycling of proteins or proteostasis, and what's the effect of proteostasis on senescence? And then,
Hmm.
let's throw in DNA damage, and sort of how does it all play with each other?
Yeah, yeah, it's interesting. So you mentioned a little bit around some of the biomarkers you like to look at for like DNA damage. Um, you mentioned functional biomarkers like VO2 max or grip strength.
Mm-hmm.
Um, so, kind of for you right now, when you look at, you know, if let's take it from bench to clinic. If you're talking to somebody and they're saying, but I really want to understand like what's my trajectory, like how am I aging right now? Um, what do you think? Where are we at with the state of the art where you would say, well, this is a biomarker I would pay attention to?
Yeah, I think always really looking at the clinical markers that everybody uses in their metabolic panels is the first place to look. Those are really functional metrics that we know we have tons of data on. You have years and years of data there. A lot of these molecular biomarkers that are related to aging that we're using, they're more recent. I know there's a really big push in the field to look at epigenetic changes with age. And so this relies upon really looking at methylation pattern changes. So you have these things called CPG islands that become methylated sort of randomly and with purpose, either in health or disease or age. And so people are using those to sort of track your biological age as opposed to your chronological age. So really, and going back to that concept, chronological age is the number on your driver's license. Biological age is sort of how you're doing relative to that age. And so some of these tests are really they're quite varied in what they find, and like we were previously talking about, it all really depends on how these tests were created and what data sets they were trained on and what endpoints of how they want to look at it. Like I said, some of these clocks could be using the odometer method of, you know, you have a 1992 Honda Civic and you do something nice to it and now it looks younger, but is it really just a really nice looking 1992 Honda Civic? Or the other way around it is, they're looking at the speedometer of aging. Is it going faster? Is it going slower? And there's some of these that do that where there is a epigenetic test called the pace of age.
Yeah. I'm curious. I I've looked a little bit at the pace of age. I've looked a little bit at kind of the I think, is it the grim age,
And pheno age, yeah.
pheno age. There's a new one that I read. It's a preprint paper on called EMR age
Hmm.
that came out of Mass General, and I just want to help people understand how these clocks are kind of made. And so I'll give you my kind of simplistic view on it, and then you can kind of correct me where I'm wrong. So as I understand it, there's different data sets of kind of longitudinal data that we have on people, meaning we have multiple timestamps of biologic data, whether that's, a comprehensive metabolic panel, a CBC, whatever. And what people like Dr. Horvath, Dr. Levine, what they've done is they've taken these big data sets, and one of those is the national data set, the NHANES data set.
Hmm.
And they took that and they found a series of biomarkers that, if you look at the levels of those biomarkers and you correlate them, you can tell how long, or predict how long somebody's going to live with some sort of confidence, like 90% confidence that, if you took some random person's labs and you looked at them, you could say, okay, this is your biological age based on these. And that's kind of the first thing they do. But then they take that data and they take samples, DNA samples from a bunch of those people, and they look at this methylation that you talked about. So these methyl groups on DNA that are usually they're turning a gene off or on, and there's like millions of these methyl sites, but they map only, a few hundred of these methyl sites or
Hmm.
something. And then I can take a blood prick and send it into a lab, and they'll look at my methylation, and it's kind of this like looking backwards. Okay, your methyls look like these methyls, and those look like these blood tests from this cohort of people, and that's kind of how the full circle goes in my head. But I'd love for you to like correct anything that I got wrong, um,
Nope,
or if
that's
you can
spot on.
simplify it.
That's spot on. So there's a really talented scientist across the street from me in the School of Public Health, Dan Belsky. And so Dan has created one of these clocks, Dunedin Pace.
Yeah.
And so what Dan has done is use longitudinal sampling of this cohort that was born in Dunedin, New Zealand, and they sort of profile them every 5 to 10 years. And Dan was doing blood biomarkers and correlating it to the physical function, and then of course doing the epigenetic clocks. But instead of sort of looking at the odometer method, he's going the speedometer method of looking at their pace of aging. And that's what Dunedin Pace sort of came out of. So really, they're all kind of doing the same things. It's just how are they training their clocks and on what particular data set. And, it's being, like you said, done on large scale data. But so there's a lot of people in those data sets, but, do any of them look like Jared or do any of them look like Matt? Right. So,
Yeah. Yeah. So I'm curious, could you make the argument for me, like, why would somebody want to use a methylation clock rather than just the biomarkers that went into training the methylation clock?
I think in some ways it can be a good adjunctive, or it's a it could be a more cost effective or rapid way to do the analysis versus sort of all this in-depth work. And two, a lot of these epigenetic clocks just require a little bit of blood on a spot card versus sort of, the good old tube of blood, right? Or multiple tubes of blood. So
Yeah.
there's also the benefit of that. They can be, less blood or less invasive, or just utilize existing blood that was collected for another purpose.
Yeah. And one interesting thing that I noticed in the newer EMR age was half the biomarkers were things I'd never drawn as a physician. Like they were
Mhm.
like I had never, some of them I'd never even heard of. And I imagine as they continue to work on these clocks, that helps the proprietary nature of these clocks. Um,
Sure.
it, I mean, I like to assume the best, like those were the exact biomarkers they had to use to get the highest, confidence.
Mhm.
But also I realize like this stuff, it costs money to develop this and you want to, protect your IP when you can.
Yeah, I think that's another push in the field is there's groups that they want to build the best biological aging biomarker, and probably to do it they need to commercialize it, so they have to have some intellectual property over it. Other ways are looking at how do we sort of democratize biomarkers so that somebody could measure a biomarker in Boston, Buenos Aires, or Baghdad, right? Like,
Yep.
and and collect samples. And how's the easiest way to do it? And it's at a reasonable price. And so maybe it's not the best biomarker, but it's good enough.
Yeah.
And so I think those are things that the field does wrestle with that too. Of how do we build the best biomarker, but is that biomarker going to be wide scale distributed?
Right. And I'd love to hear, like, I kind of have in my head why
Mhm.
aging clock would be useful or why biomarkers of aging are useful, but kind of how do you think about it? Like, why why is there this quest for biomarkers for aging? What are we looking for?
So I think first of all, we want to use them, like I said, to measure our biological age relative to our chronological age. Sort of how are we doing for our age? Are we doing poorly? Are we doing fantastic? And two, uh, in response to interventions, whether they be drug or lifestyle, of, hey, I started doing caloric restriction or intermittent fasting or time restricted feeding. Are my biomarkers moving in a positive direction? Is the juice worth the squeeze? Like, am I getting payoff for what I'm doing? Or people are starting to do some of these clinical trials that are sort of there for age related chronic diseases. So people are on rapamycin. Are their biomarkers changing in response to rapamycin, as well as sort of their physical function metrics? So it's just it's layering the confidence onto the data that we're getting. The
Yeah.
more data you have, the better it is. But then again, it's also the other thing of, if your biomarker has 50 parameters, is it better than a biomarker that has 40 parameters? Or is it just basically both of your biomarkers stink if it takes that many things to like show something?
Yeah, and one thing that's interesting to me about kind of aging is because, as we talked about, there are these multiple hallmarks, and we think that it's kind of affecting the whole organism. And so, what's really interesting to me is, can we find things that will affect the whole organism in the opposite direction? And kind
Mhm.
of like you said, like if I have somebody doing zone 5 exercises twice a week, like personally I hate zone 5. Like zone 5 is the worst. Every time I do it, I'm just like, oh, why am I doing this? But if I had something that I could check, and I have VO2 max, and that's what I
Mhm.
use today, and VO2 max is predictive and tells me mortality risks and things like that. But if I had a blood test also that said, hey, when you adopted VO2 max, look at how your rate of aging or your biological aging changed, like that's super interesting to me.
Yeah, there's for us in the senescence field, like we were talking about the inflammatory factors they produce, there's a blood test for one of these factors called GDF15, growth differentiation factor 15. And what it's been shown is to increase in circulation with biological age or disease. And there was a recent study that showed that it's actually a good predictor for risk of death.
Hmm.
So, if you're doing all your zone 5 exercises and you're looking longitudinally and you see by doing this your rates of GDF15 are going down, you feel better about doing it, right? You don't, maybe it
Oh,
doesn't generate as much anxiety before you go do it.
yeah, totally, totally. And I mean, I definitely am interested in like biomarkers like that, like very
Mhm.
predictive biomarkers. Um, I'm curious, like, so there are some biomarkers that we talked about, aging clocks, GDF15. What other biomarkers are you kind of interested in looking at right now, or what are the biomarkers out there that you're like, oh, this is one we need to continue to learn about?
So I like to look at DNA damage. There's a form, one of the most common forms of DNA damage is adeoxyguanine, and it's basically oxidative damage of guanine or the G in DNA. And that's been commonly used for exposure studies, but we know that accumulates with age as well too. Another thing I like to look at is mitochondrial DNA that's sort of cell free. Like mitochondrial DNA definitely has a purpose for being inside the mitochondria inside your cell. When you find mitochondrial DNA outside of the cell, it's typically because the cell has broken open and released it, and it can be inflammatory.
Mm.
And it's been shown to be a good biomarker for a number of different diseases and conditions. So it's one that I'm sort of interested in looking at. And, can we sample it in the blood or can we sample it in some other biospecimen? And jokingly, sort of one way the lab is looking at it is, of course, through, the good old blood tube. And the other way we're really starting to look at it is through the good old biospecimen cup where, you could get urine samples. And so it sort of be even more non invasive than sort of a less invasive blood draw, and correlate mitochondrial DNA to nuclear DNA in the urine and see, is it are the rates increasing as we age? Are they decreasing with interventions that we're doing?
Yep.
So, yeah.
Yeah. When you think about the current emphasis of, you know, different therapies that people are testing, so the TAME trial where they're testing metformin, you know, there are some smaller trials with rapamycin and
Mhm.
some kind of non-blinded studies and things like that. Um, what are some of the areas that kind of pique your interest? What are some of the things that are ongoing that you're most interested to see outcomes of?
I think we've seen some outcomes from one of the trials that really excites me is a study called the Calorie Study, Calerie. And it was looking at caloric restrictions in humans over 2 years. And the goal was to calorically restrict by 25%. I think effectively they got to around 12%, but they saw metabolic improvement. And what they've seen is a number of people have been doing biomarker analysis. And so their epigenetic clocks improved alongside their metabolic pathways. So you increase insulin sensitivity. They saw a reduction in that age related inflammatory molecules like GDF15 and other molecules that could be associated with senescence or just generalized inflammation. Um, so there's a number of really good biomarker studies that are being done with that because it was a nice data set that was done over 2 years. And that's to me, one of the easiest interventions. It's not even a drug or, something else.
Yeah,
Otherwise, there are studies with rapamycin here at Columbia. There's the Vibrant study, which is looking at rapamycin and ovarian aging.
yeah, yeah. The I'm curious. Um, so caloric restriction, we've known for a long time that you know it's really good outcomes in
Yeah.
mice in the ITP. Um, do you and I guess where I'm going with this question is I feel like we go through trends in aging where we get super interested in something and for a while everybody was taking metformin. I was running into like 30-year-olds who were taking metformin because they heard that metformin was going to keep them young forever. NMN has had its moment. NR, some of these may prove to be beneficial. The ITP has done studies on some of these.
Mhm.
Um, do you are there any right now that are not top of mind for people that you think are interesting to you? So like I named rapamycin,
Mhm.
metformin, NR, NMN, resveratrol, like those types of things. Those are the ones that people have thought about, heard about. Are there any that you're interested in that aren't top of mind?
So, yeah, I'm inherently biased because I work in this field, but the field of senolytics, so drugs that either selectively eliminate senescent cells or reduce their inflammatory effects or phenotype is what we call it. Those are interesting. Drugs that regulate mitophagy, so it's sort of improving mitochondrial function. And I think all these drugs, we all have to go back to the biomarkers. Does somebody need to be on metformin at that age? Similar, like, does somebody need to be on a senolytic? Well, we should be testing for their senescent cell burden first, because if they don't have a senescent cell burden, there's no reason to treat them.
Hmm.
Or somebody that wants NAD. Well, do you have low NAD?
Right.
So yeah, I think that's sort of a driving case of all these different interventions. What's Are there deficiencies there? And we can figure that out from the biomarkers. Or if we're treating with NAD, we can also use biomarkers to look at DNA damage or markers of senescence to see sort of what are the wide and varied effects of that drug, not only on sort of its intended pathways, but the other ones. Are you sort of getting off target biological benefits?
Hmm. Do you one thing that I think gets a lot of airtime is exercise.
Mhm.
Um, are there any good studies that show that exercise actually helps with like hallmarks of aging?
So I was part of a study that was led out of the Mayo Clinic, and the Mayo Clinic had a elderly population of healthy elderly individuals that they were put on progressive resistance exercise training, a very sort of brief course of it. And what they found were not only did their physical function parameters, like sort of get up and go, walk times, uh, increase, or sorry, excuse me, benefit. But what they also saw was a reduction in senescence markers in their blood, as well as the circulating inflammation. It was kind of one of the first studies that tied senescence biomarkers to a human intervention study from a lifestyle intervention perspective, and it worked.
Yeah, that's super interesting. I mean, I felt kind of silly asking that question because there's
Mhm.
so many studies that show like if you increase aerobic capacity, then all-cause mortality goes down. If you increase strength, all-cause mortality goes down. But it's interesting to hear that you were actually able in that study to show both some sort of biomarker improvement, senescent cell load decrease, as well as increased function.
Similarly, in the calorie study, there's a recent publication that came out that showed that in the adipose of those calorically restricted individuals, they saw a reduction in biomarkers of senescence.
Hmm.
And really, adipose tissue can be a hotspot for senescent cells. So if anywhere was going to show an improvement, it should be there, and it did. So
Hmm.
really, we're starting to hit with all these studies going on and sort of the validation that we've done with these biomarkers. The rubber's hitting the road now, and it's
Yeah,
kind of nice to see it.
yeah. I'm curious. I feel like somebody could spend almost an unlimited amount of money. And when I see people like Brian Johnson, he does spend an unlimited amount of money on this type of stuff. Like if you personally were like saying, okay, well, I want to live as healthy as long as I can, what would you do outside of, eating right, exercising, sleeping good? Like or and some of that you don't have to go into like personal like habits or anything, but like, as people think through this, where do they decide, or what are some of the things where they might decide like this is worth a little extra effort or money, supplement, testing, etc.?
Yeah, I think you know, for me, it would be routinely looking at metabolic panels or some of these physical function metrics, because we know those are definitely involved in sort of our health and or decline of our health. I think another thing that people don't look at enough and really has a profound effect is social stress. Social stress has a massive effect on aging and can premature like that. I've read studies that social stress can prematurely age the immune system. So, and we've seen it with social stress models that it will age the study animals.
Right.
So, and shorten their lifespan. So, I think that's a huge effect of you're working, so hard physically. Are you doing it from sort of a mental approach as well? And that could be not even counseling, but just the relationships that you have with people and the sort of the social effects of that.
Yeah. I mean, the Surgeon General came out with his loneliness book,
Mhm.
and there's this concern that we're becoming a more and more lonely society and not having true relationships or community. And it's interesting to think that that's probably making us age faster. Um, and it's interesting when you look at blue zones, it seems like
Mhm.
community and relationships is a big part of that increased longevity in those areas of the world. And so it's interesting to think like you can actually see biomarkers of like social stress equals more senescence,
Yes,
equals
yes.
more DNA damage, or whatever it is. Like, that's super
That
interesting.
happens. Yeah. And so it's kind of, to me, the easiest things that we can do. Well, the easiest thing is obviously genetics, right? You have it or you don't, or you have your modifiers. You didn't do anything to get it, you know. And from there, it's diet and exercise.
Hmm.
Those continued work with that can have profound impacts. And then from there, looking at biomarkers and understanding areas where you may be sort of pushing on the gas pedal of aging or ways that you could slow it down from there and let the biomarkers or the clinical markers guide you.
Right.
And also use them as surveillance to see if your intervention is having an effect.
Right. Yeah. If you're putting effort, money, whatever it
Mm-hmm.
is in, you want to know if it's working.
Sure.
So yeah, I've been reading Nir Barzilai's book about his study on the Ashkenazi community up in New York
Mm-hmm.
City, and like you said, genes is the easiest way. Then I can smoke and eat whatever I want and live to 104. Um, but I think what I think is interesting about this space is not only can you spend a ton of money on um, biomarkers to see what's happening, you can spend a ton of money on supplements
Mm-hmm.
and things like that. And what I didn't hear from you was, okay, here are the 20 supplements you should be taking today to make it so you don't age as fast. Um, and I I think that's reassuring that
Yeah.
really it's diet, it's exercise. I do think the ITP has been interesting to watch, but they're still mice. Like, I have to keep
Yeah.
reminding people like they're not humans, and some of those are really safe drugs. That probably it's just fine for people to take if they want, but curious how you think about kind of the ITP and outcomes from there, and if somebody you knew, if a family member came to you and said, "Hey, I'm thinking of starting on meclizine. What do you think?" Um,
Yeah. So I think the ITP is really interesting. I think they measure lifespan. They don't really look at health span, which I think is really becoming the goal of the field, or for many of us in the field. And health span, I should back up and say, is this period of disease-free survival. It's sort of how well do you live for how long you can live, and then lifespan is just the terminal end. And ITP only measures lifespan, and they're doing it with drugs that either can be given compounded into the chow or put into the water. So anything that would be dosed by injection, those don't make it into that study. And it's done in a unique strain of mice. So there's sort of all these different strains of mice. But there's a lot of stuff that's worked, like caloric restriction, rapamycin. The ITP has also shown us a lot of these drugs have sex-specific effects where they may only
Mhm.
work in a male mouse or a female mouse. So that's interesting. But Rapa does seem to work in both male and females in the mice. I believe it's a different concentration of the drug in the chow. Caloric restriction broadly works across both sexes. There's no real
Yeah.
effect to that, and it's dose dependent. The more you calorically restrict, the more of a benefit you see up to a certain percent. Above sort of 50%, it starts to plateau, at least in mice. So
Yeah.
don't go overboard. And some people have done the combination of caloric restriction and time restricted feeding, and that's
Mhm.
had a stronger effect as well too with the animals. So I think
Yeah.
that's it's really cool stuff to see, and I think they can do large scale studies, and it's really nice because it's actually done at three different sites. So the University of Michigan, the Jackson Laboratory in Bar Harbor, Maine, and the Barshop Institute down in San Antonio, Texas. So really three different environments, and you'll see there are some differences in lifespans in the individual sites. So not
Right.
don't just look at the composite lifespan, but look at the individual ones. But what they're seeing is really sort of robust changes. And
Yeah.
so I think the ITP is one of the gold standards that we use.
Yeah. I heard that mice like it in Michigan. They live longer in Michigan for some reason on
Yeah.
average. So, calorie restriction is this interesting thing because it's so hard for humans
Hmm.
in our current environment. So, I mean, if we told people, and I mean, I've had this discussion with people I love, my maybe some family members where
Hmm.
I'm like, hey, if you lost 20 pounds, you'd live longer. And they're like, yeah, but I'm going to eat my cheeseburger and my shake and live as long as I live. Like I want to enjoy life. And the other thing that I think is interesting around calorie restriction is just muscle mass.
Hmm.
And as of late, there's been multiple books about, you really need to go into old aging with as much muscle as possible. And if you calorie restrict, you're not going to have as much muscle as you could have. And do you feel like there's anything in the way of science today that we can point to and say, yes, you should build as much muscle as possible and not calorie restrict, or you should calorie restrict more and yes, exercise and try to maintain strength, but not care as much about muscle mass?
So that's a good question. I sort of don't know that I feel comfortable to tackle that. I will say this, that there's been a lot of work being done by Dudley Laming's group at the University of Wisconsin on branch chain amino acids and actually their restriction having elongating effect on lifespan.
Yeah.
So, like reduction of isoleucine extends lifespan. But you have to remember, mice and by and large are very sedentary animals.
Hmm.
So I think once again we get back to that thing that mice are a good model, but mice aren't always people.
Right. Yeah. Yeah. And I have a hard time, like, if I think of the phenotype, so the way
Hmm.
people look and act, like, somebody who's 90 who can, kind of walk really fast and they have a steady gait, and it's because they have muscle mass and strength, like, and somebody who doesn't, like, and we know that sarcopenia, loss of
Yeah,
muscle mass with age is a huge deal and
yeah.
leads to, and like you said, health span is what's really important, not just lifespan. Like the
Hmm.
last thing I want to do is be, in a bed for the last 20 years of my life.
Yeah.
Um, so it is interesting to think through those trade-offs.
But you're right. You think that lean mass is a good thing. As you sort of head into aging, probably having a decent amount is great because sort of for the rest of your life, to some degree, you start to play defense and just try to keep what you have, right?
Yep. Yeah, exactly. it's kind of like trying to project ahead and say, well, I want to have strength to do certain things. I'm going to have to train right now so that I have enough strength
Yeah.
when I get there.
You sort of, saving for retirement, but, instead of dollars, it's muscle mass.
That's right. That's exactly right. So Matt, this has been great. Any last thoughts before we kind of finish up? Anything that we didn't cover that's been kind of top of mind for you in the aging area lately?
I think it's just a really dynamic feel. So kind of always keep an eye out, and there's a lot of things that come into play that may go back out. There's for a period of time, the field got really excited about telomere links, sort of, the ends
Mhm.
of our DNA. And we always use the analogy, it's sort of the plastic end on the bit of your shoelace and how those erode. And that was sort of a driver of aging. And it can be in some ways, and it can be that for particular cell types, cells that are always proliferating. But a lot of cells in our body are what we call terminally differentiated, so they're no longer proliferating. And telomeric erosion isn't a big thing for that. So, telomeres were used as a biomarker for a long time, and we kind of found out they weren't really that responsive to interventions. And so I think it's sometimes that really we're doing biomarker refinement. We're finding things, and ideally we're making better biomarkers or figuring out in context where they can be used or where they're more applicable. So I
Yeah.
think, it's not a static field, it's dynamic. So just kind of pay attention if you can.
Well, I certainly appreciate your guidance and your help in this space. I mean, it's huge for me to have somebody who's looking at this day in, day out, because there is so much noise.
Mhm.
And actually finding like what is the true signal inside of all this noise, and like like you kind of said it in the end, like it's, exercise, eat a good diet, pay attention to the science. There might be biomarkers that come down the line, but right now do metabolic panels, look at kind of hemoglobin A1C or metabolic health, those types of things. Like that seems like that's kind of the key for now, and we'll stay tuned for more exciting news coming from you and other scientists.
Yeah, the new stuff, it can be exciting, it can work, but the old stuff works because it works, right? So
And it's usually much less expensive than the new stuff.
that too.
So Matt, thank you so much. This has been great.
Yeah, thank you for having me. This has been awesome.